Welcome. So this assignment deals on studying the intestinal paraglycoprotein, which is exporting endocannabinoids to prevent inflammation and maintains homeostasis. So the major research question here is there any potential for the paraglycoprotein mediated secretion of this N-acetylethanolamin type of endocannabinoids to functionally suppress this hypoxylin A3 mediated intestinal inflammation in vivo? So the hypothesis that was made is the CB2 deficient mice is less able to respond to these paraglycoprotein secreted inhibitory type of endocannabinoid. So the basic thing for this research is that there is two kinds of migration for the neutrophils to enter from the serosa to the lumen. So first, for the cannabinoids, endocannabinoids, or for the neutrophils to enter from the serosa to the lamina propria, we will be having a chemoattractant like IL-8, interleukin 8, which is released by the basolateral surface of epithelial cells. So this is the first step here. And the second step is the migration of neutrophils from the lamina propria to the lumen, which is facilitated by eicosanoid hypoxylin A3, which is usually released by apical surface recorded by the MRP2 protein in the epithelial cells. And the another point here is that this HXA3 is usually inhibited by a protein called paraglycoprotein, which is usually secreted by a gene called MDR1 gene. So this HXA3 is highly lipophilic in nature. And the design of this experiment is in such a way that first the scientists have analyzed the paraglycoprotein, which is a secreted lipidome of normal cells, to identify lipids capable of inhibiting this HXA3. Then they confirm that there's something they call as amend, a group of lipids that is secreted by these paraglycoproteins, and they decipher its chemical composition. Then they confirm that only this N-acetyl-ethanolamine type of endocannabinoids, which is a part of this amine, is gonna inhibit the HXA3 mediated neutrophil migration. Then they decipher that CB2 receptor is a target on immune cells for various type of ECs. Finally, coming to the point that this paraglycoprotein mediated secretion of NAE type of endocannabinoids functionally suppress the HXA3 mediated neutrophil migration in vivo. So as per the question given in the assignment, the figure 6 is most matching with the goal of the experiment, that is point 0.5 here. So coming to the figure 6, aspects of figure 6, diagrams A, B, and C. From this, what we are able to infer is that the CNR2 minus negative mice treated with this DSS were more vulnerable to inflammation, and they were able to develop increased intestinal pathology and weight loss compared with the wild type mice. And also there was a dramatic increase in the neutrophil influx into the intestinal lumen, like which we are able to see in figure C here, and which is graphically shown and observed from figure A and B. We are able to see that there is a dramatic decrease in weight, that is shown in figure A, and the histopathology score has been increased in case of the CNR2 minus mice. And here in figure D and E, we are able to infer that the presence of neutrophil infiltration in intestine using assessed using myeloperoxidase assay from fecal samples showed us that the CNR2 minus mice is displaying a dramatic increase in the neutrophil influx in intestinal lumen, while there was no such significant change in the neutrophil levels in case of normal tissues. And this was also extended to this figure F, G, and H, from where we are able to see that CNR2 minus mice does not exhibit significant change in tissue neutrophil count, both in case of absolute number as shown in figure F and in case of percentage shown in figure G during the flow cytometry analysis. Similarly, the FACS analysis of the lamina propria for neutrophil infiltration also revealed there was no big significant change. So that is what is evident here. And finally, like coming to figure I, J, and K, to confirm, the increased susceptibility of CNR2 minus mice to intestinal inflammation resulted from a failure of neutrophils to respond to this endocannabinoid inhibition. Bone marrow chimeras were created, and the relative contributions of the CB2 in the immune and non-immune compartments to susceptibility to this DSS-induced colitis was evaluated. And what was found from this was, overall, the mice transplanted with the CNR2 minus bone marrow, regardless of its genotype, we are able to see that there is a display of greater weight loss from figure I, and colon shortening in case of figure J, which is indicating the increased inflammation, that is neutrophil infiltration. Similarly, transfer of this wild type bone marrow into a CNR2 minus recipient partially abrogated the inflammatory phenotype, and these heterologous mice developed less inflammation than seen in the CNR2 minus bone marrow donors into the CNR2 minus recipients. So, based on all these, we are able to conclude that we have defined an anti inflammatory paraglycoprotein endocannabinoid pathway that is acting to counterbalance the pro inflammatory MRP2 HXA3 axis. We propose that balancing the paraglycoprotein endocannabinoid and MRP2 HXA3 axis is a mechanism by which epithelial cells themselves integrate signals from the local environment to maintain exquisite control over the initiation of neutrophil mediated inflammation. Thank you.